Okay, let's start the session. I hope I'm audible to all of you.
Yes, ma'am.
Yeah. Hi everyone. I'm Lavanya, a PMR research scholar from IIT Madras. Today I will be conducting a live session based on the course contents of the course Tissue Engineering. So the live session will be about scaffolds, ECM, natural and synthetic polymers. So let's go through the week 2 course video contents. We would have seen about scaffolds, its role in tissue engineering, desired products. So scaffolds are basically for cell supporting growth, cell adhesion, biocompatible and biodegradable in nature, and will provide mechanical properties for the tissue to grow. You would have seen about different scaffold materials, the basic extracellular matrix and its components, decellularization and various decellularization techniques, various natural based products, that is alginate, dextran, chitosan, cellulose, hyaluronan or hyaluronic acid, silk fibroin and so on, and various synthetic polymers for scaffold fabrication like PCL, PLA, PLGA, PEG and so on. And you would have also seen about the techniques used for scaffold fabrications. Finally, hydrogels, types of hydrogels and cross-linking techniques. So this is an overview of the week 2 video contents that you guys would have seen. Now let's look at some questions that will help you understand the week 2 contents better and approach the week 2 assignment. Okay, you can stop me anytime if you're having any doubts. I won't be able to see the chat because I'm in slideshow, so you can unmute yourself and you can ask questions. Let's start with the first question. A 49-year-old patient with severe burn injuries to the skin on his right hand is brought into an emergency room. Suggest the best treatment option that can be given to the patient from the list of options given below. So if there is a severe burn injury patient that has been brought into the emergency room, what treatment option would you suggest from the options given below?
Ma'am, autograph.
Yeah, like autograft is the correct. That's correct. Autograft is one of the main approach that will be followed for burn injury patients. Isograft, xenograft, and other options are not applicable for this question. So the answer is A, autograft. It has very less chances of immune rejection, and it is the immediate option that will be followed for treatment of burn injury patients. Second question, which of the following ceramics would not promote bone ingrowth? So you guys would have studied about bone ingrowth through a material, biomaterial. So the question is, which of the following ceramics would not promote bone ingrowth? So there is more than one option here. Zirconia, calcium oxide, phosphorus pentoxide, alumina, zirconia alumina complexes. Yeah, can anyone of you answer this? Okay, the answer is zirconia alumina and zirconia alumina complexes. Why? Because so usually for bone ingrowth, the biomaterial should be bioactive and not bioinert. So zirconia alumina or zirconia alumina complexes are bioinert biomaterials. So for bone tissue engineering, with regard to bone tissue engineering, you guys would have studied about two types of materials called bioinert materials and bioactive materials. So bioinert materials will be used for implants which does not require any bone to be grown through the material. But bioactive materials are materials that are used for bone growth through the material, and eventually the material by itself will degrade and resorb, which will give space for new bone formation. So these are the two types of material, bioinert and bioactive materials. So the answer here is zirconia alumina and zirconia alumina complexes, which would not promote bone ingrowth since they are bioinert materials. Calcium phosphate, the third question is calcium phosphate bioceramics such as hydroxyapatite and tricalcium phosphate that promote bone ingrowth are called as dash. So the answer for this question is bioactive ceramic. So biomaterials, you would have seen the classification of biomaterials as polymers, ceramics, carbons, and so on. So ceramic is one type of biomaterial that is usually used for bone tissue engineering applications. And here calcium phosphate bioceramics such as hydroxyapatite or tricalcium phosphates are bioactive ceramics that are used to promote bone ingrowth. So for a tissue repair, this picture is an example for bioinert and bioresorbable or bioactive ceramics. So bioinert ceramics like these I said, zirconia or alumina or titanium are used as implantable materials. So for implantable materials, you will not want bone ingrowth. So if there is a handicapped patient and you want to implant any material into his tissue, you will know that there won't be any new tissue growth happening, and it shouldn't support new tissue growth also, because the tissue there is a tissue growth there has been stopped. So you will need a bioinert material there which is non-degradable, which has stress shielding characteristic, which has low biological response, low bone resorption, and no bone ingrowth. Opposite to that, bioresorbable or bioactive materials, which are made of magnesium and zinc, will have bioactive compounds, will be naturally bioactive, and the degradation of those materials depend on the local environment. So as and when the bone is growing through the biomaterial, slowly the biomaterial will start degrading. It will have high biological response also. Any doubts?
No, ma'am.
Yeah, okay. So the next question, choose a cellular tissue engineering product that has been commercialized. So given below are some commercial tissue engineering products available. Like in the last session, we spoke about Matrigel, and along with Matrigel, Dermagraft, OP1 putty, and Alloderm. So these are some of the commercialized tissue engineering products that are available. Among these, which one is a cellular tissue engineering product? That is, it contains live cells. Okay, the answer is Dermagraft. You would have heard about this in Sir's lectures. So Dermagraft is a skin tissue engineering product. So this Dermagraft, this is the product, and this Dermagraft is a material that is made up of polyglactin 910. So polyglactin is nothing but PLGA. You would have heard about PLGA, a synthetic polymer, in Sir's lectures. That is polylactide glycolide acid, PLGA polymer. So PLGA polymer will exist in different ratios. That is, different ratios of PLA and PGA combined together will form PLGA. So 90% of PLA and 10% of PGA will combine together to form polyglactin. So this is the synthetic polymer that is being used as a base in this particular commercial product, Dermagraft. And this is called a cellular tissue engineering product because it contains neonatal allogenic fibroblast cells. So we also saw about fibroblast cells in the last session. Fibroblast cells or keratinocyte cells, both of these cells are invariably used in this particular graft to produce this product. So this product is majorly used for chronic diabetic wound healing applications. So you would have heard about diabetic wounds. Diabetic wounds will take a more longer time to cure compared to normal wounds, and these diabetic wounds are almost always chronic. So you need a graft that will be able to initiate the wound healing as well as skin regeneration in that particular area for it to heal completely. So this particular derma graft is a cellular tissue engineering product which will initiate help in wound healing as well as tissue regeneration. So let's move on to the next question. What are the components required to engineer a functional tissue? So given below are some options, among which you have to select the two options that contains the components that are required essentially to engineer a functional tissue. So what might be the two options? Option A, biomaterial.
Ma'am, it is A or option A and option B.
Yeah, that's right. So option A, biomaterials, cells supporting growth factors, and option D, stem cells, scaffold, and recombinant proteins. So these two together are required to engineer a functional tissue. So you will not need microorganisms to prepare a tissue because microorganisms are considered as pathogens here. Cells and supportive growth factors alone are also not sufficient. So the answer is option A and D. Okay, the next question. Which of the following includes functions of scaffold? Option A, support the cells and regulate their function. Option B, direct the cell proliferation only when they are seeded with cells. Option C, cell delivery depot. Option D, helps in teratoma formation. So which of the following includes functions of a scaffold?
A and C.
Yeah, exactly. So it supports cells and regulate their function. Direct the cell proliferation only when they are seeded with cells. So that is not the case with scaffolds. Even when they are not seeded with cells, even if you just place a empty scaffold in your body, it will help in cell migration and proliferation and thereby production of the new tissue. So you need not have cells seeded in scaffolds always. Third option, cell delivery depot. Yeah, it will act as a cell delivery depot as it helps in cell proliferation and migration and helps in teratoma formation. So it will never help in a teratoma formation. A scaffold which helps in teratoma formation is not at all a biomaterial that will be used for tissue engineering. So the answer is option A and C. So the next question, decellularized extracellular matrix is dash. Immunogenic, non-immunogenic, malignant, non-biocompatible. So you would have studied about decellularized ECM in your first week 2 video. So
Is B the option?
yeah, B is option. So decellularized ECMs should be non-immunogenic. It can't be immunogenic or malignant or non-biocompatible. Decellularized ECMs are basically tissues, live tissues that has cells which are taken from cadaveric animals, and they will be decellularized using some detergents, and they will be used, the scaffold, the ECM, the remaining ECM will be as such used for growth of other human cells, and finally to produce a functioning tissue. So decellularized ECM is used for that purpose. So the answer is option B. Any doubts?
No, ma'am.
Yeah, okay. Okay, moving on to the next question. X is a mucopolysaccharide that is a vital component of the ECM. X helps in maintaining the fluidity and mechanical strength of the ECM. What is X? So, select from the options below: glycosaminoglycan, starch, silk fibrin, or elastin. So, one main hint here is X is a mucopolysaccharide. Mucopolysaccharides are polymers that are present in the fluid component in our body. So wherever mucus or fluid components are present in our body, there this polysaccharide will be there.
Ma'am, first option.
Yeah, glycosaminoglycan. That's right. So one main example that I can give you is the articular cartilage. So in our body, we have various joints, and one main joint is the knee joint. So the knee joint will have this fluid called synovial fluid. You would have all heard about it. Synovial fluid, which will maintain the fluidity and the mechanical strength of the joint area present there. So main components that are present in this synovial fluid are glycosaminoglycans. So the answer is A, and I have listed here some of the glycosaminoglycans that are present in various parts, various tissues in our body. So hyaluronic acid or hyaluronan that you would have studied is present in skin, synovial fluid, bone, cartilage, which is humor, and so on. Chondroitin sulfate, which is present in cartilage, bone, and CNS. Keratin sulfate 1 and 2, which is present in cornea, cartilage, and lucidity. Heparin, which is an anticoagulant that is about which we studied in the last lecture also, is present in mast cells, liver, lung, and skin. And then heparin sulfate, which is present in skin and kidney. So glycosaminoglycans can be divided into further divided, characterized into two types, that is polymers which are sulfate based and which are non-sulfate based. So hyaluronic acid is a glycosaminoglycan which is non-sulfate based, and chondroitin, heparin, keratin sulfates are glycosaminoglycans which are sulfate based. So these are some examples of mucopolysaccharides. Moving on to the next question. The formation of pores due to the process of freezing and sublimation is dash. Condensation, lyophilization, solvent casting, and gas forming.
Lyophilization.
Yeah, it's lyophilization. So lyophilization is one particular process that is that will help in formation of scaffold, and it is very easily used almost in all of the experiments that are being conducted now for scaffold formation. So the answer is option B. Lyophilization is a process where you prepare a scaffold, like you prepare a pre-polymer mix, and when you keep it in a freeze dryer, so freeze dryer is also called as lyophilizer. Yeah, freeze dryer is also called as a lyophilizer. In a lyophilizer, when you place your pre-polymer mix, due to the low temperature and low pressure conditions, that is, in these conditions, it will directly be converted from a solid to a vapor or from a liquid to a vapor. So conversion of solid to vapor directly is called sublimation. So this is how scaffolds are formed. So when your pre-polymer mix, whether it is a gel or a liquid, it will have lots of water molecules locked into it. when you are placing it in a freeze dryer, the water molecules will be directly converted to vapor and giving you just the outer scaffold. That will be directly used as a substitute for ECM, and cells will be seeded onto it. Moving on to the next question, what is or are the importance of crosslinks in a hydrogel? So, you would have studied about what a hydrogel is. Hydrogels are 3D constructs that will hold water, but it is hydrophilic, but it is not soluble in water. So, the not being soluble in water part of a hydrogel is mainly due to the cross-linking that is happening between polymers to form a hydrogel. So, what is or are the importance of cross-links in a hydrogel? Options: add stability, interconnects polymeric networks within the hydrogel system, induces gelation, helps in disintegration of hydrogel. So, what could be the possible answers?
A.
Yeah, A is correct. It adds stability. Along with A?
A, B and C.
Yeah, that's right. It adds stability. It interconnects the polymeric networks within the hydrogel system. So, for example, if you're using a polymer called alginate and a polymer called gelatin, and if you are preparing a hydrogel out of it, it will connect both these polymeric systems together and will give you as one single polymer, and that will be a one single polymer. It might be in a liquid form also. So to convert it into a gel form, to induce gelation, you will need extra cross-linkers. So even without cross-linkers, if there are particular functional groups, for example, if there is an amine functional group, yeah, so take this P1 as polymer 1, and it is having an amine functional group, and take this P2 as polymer 2, and it is having an acid functional group. So these two are functional groups that has the potential to react and form a bond, NHCO bond. So NHCO bond is nothing but a peptide bond. So this bond will be formed between two polymers. So in this case, there is no need of extra chemical cross-linkers required, because if you just activate the COOH group in polymer P2, and if you bring a primary amine near it, it will automatically form a bond. And this bond formation only will lead to a formation of a gel. So not mixing of two polymers alone is required to form a gel. You need a particular cross-linker to form a gel. It may be a physical cross-linker or a chemical cross-linker, or the polymer should have functional groups that will react together to form bonds and thereby forming a gel. So the options are option A, B and C. Helps in disintegration of hydrogel is not applicable here. So cross-linking, if you use a cross-linker and if your hydrogel is still disintegrating, then the gelation process is incomplete. So that is what it means. Yeah, so here you can see examples of some cross-linkings. So physical non-covalent cross-linkings. Yeah, physical non-covalent cross-linkings. Examples of that are hydrogen bonding, which is the bond that is simply formed between oxygen, electronegative, and and an electropositive atom, partially electropositive atom. So all these hydrogen bondings can occur anywhere where there are hydroxyl groups. So hydrogen bonding is the weakest form of cross-linking that you can rely on. It will form gels, it will definitely form gels, but since it is the weakest form of cross-linking, it won't be stable in a solution, in a salt solution for a long time. So for example, if you are preparing a hydrogel and if you are putting it in PBS, phosphate buffer saline, at 7.4 pH, which will mimic the body fluid. If you are putting it in that, it will disintegrate very easily. So it is a weak form of cross-linking. Apart from that, hydrophobic interactions are there, ionic interactions are there. One example of ionic interaction is the sodium alginate, calcium alginate gels that are formed. So calcium will replace the sodium ions and form calcium alginate gels. Have you heard of this calcium alginate gels or beads? You would have heard of it for cell immobilization or enzyme immobilization techniques.
Yes,
Yeah, that is one basic example for ionic interaction. And chemical covalent cross-linking involves Gibbs-base reaction, click reaction, and amide bond. So one example that I told here is the example for amide bond reaction. Apart from that, Gibbs-base reaction and click chemistry. You would have all known about the click chemistry, which even won the Nobel Prize. So click reaction is one particular type of irreversible cross-linking reaction. So once it is cross-linked, yeah, it's permanent. It is not reversible. And Gibbs-base reaction is a reaction that takes place between an aldehyde group and an amine group. So like an amide bond. So let me explain. So this is a reaction that is taking place between an acid group and an amine group. So similarly, when a reaction will take place, when a cross-linking will happen between an aldehyde group and an amine group, it will form an imine bond. So you would have heard of an imine bond, and this is otherwise called as Gibbs-base reaction. So imine bonds are usually pH sensitive bonds. So pH sensitive bonds in the sense when the pH of one particular area in our body or in our tissue is less, then imine bonds will be broken in that particular area. So if you want to prepare a hydrogel loaded with drugs to release in a less pH, you can incorporate imine bonds in your hydrogel and you can prepare your hydrogel so that your drug release will be based on the pH of that particular environment. If the pH is less, the drugs will release. If the hydrogel will swell, basically. The hydrogel will swell, disintegrate, and drugs will get released. If the pH is in a normal 7.4 or in a slightly alkaline form, the gels will be intact and drugs won't release. So this is an example of pH based drug release hydrogel concept. So these are different cross-linkings that we use in the biomaterials to form hydrogels. Any doubts?
no, ma'am.
Okay. So moving on to the next question. The graph represents the polymer's elastic modulus over a wide temperature range. What does the point A denote in the graph? So this is a graph between modulus, elastic modulus or Young's modulus versus temperature. So we saw I explained what a Young's modulus is in the last lecture. So can anyone say what it is? A Young's modulus or an elastic modulus? What is it dependent on? Okay, so elastic modulus is basically a ratio between the stress and the strain. Stress experienced by the material at different percentage of strain given. So the ratio between stress and strain is elastic modulus. So when an elastic modulus Elastic modulus will obviously decrease with increase in temperature. So what does this particular point A denote? Options are glass transition temperature, polymer transition temperature, melting point, boiling point. Can anyone answer?
Option B.
No, it's not polymer transition temperature. It's glass transition temperature actually. So, like I'll put it in simple words. So there is a polymer. Let's take this as polymer P1. So when a polymer is kept in a furnace and when the temperature is increased, the first transition, this will be the curve. This is how a graph of a polymer when it is kept in a furnace, the elastic modulus graph will be like this. So when the temperature increases, the first major transition that will occur in a polymer is the glass transition. So the polymer from its ductile phase will be converted into a brittle phase. So in that brittle, there is a change in its phase itself, and brittle phase is what they are referring here as glass. So a polymer will be ductile in normal temperature. It will be like foldable. You can easily fold it, and you can prepare it to your particular need. But then when it is changing based on temperature increase, it will undergo a glass transition. So it will become brittle, it will become amorphous. This particular temperature is called as CG. Another major phase change is here. So this particular temperature is called as TM, where the polymer, when you keep it in a really high temperature, it will convert itself to a semi-solid, semi-viscous, and finally a viscous material. So it will melt completely and it will have flow properties. So whatever polymer you will be using, you have to look at its glass transition temperature and melting temperature, because one practical reason that I can tell is you're preparing a material, okay, you're preparing an implant, and your implant anyway in our body, always the temperature will be maintained in 37 degree only, to the maximum 42 or 43 during fever. So beyond this, the temperature of our body won't increase or decrease. But why do we need to study about this glass transition and melting point of a polymer or a ceramic or whatever biomaterial that we use? Like, why would it be important? Any guesses? Okay, it's fine. There are only two simple reasons. One basic reason is that when you are fabricating your polymer or your implant or any biomaterial or scaffold, you will fabricate it by using you'll fabricate it at different temperatures. So for example, if you are using a polymer PVA will melt and PVA will be soluble in water only at 90 degrees Celsius. So you have to know that your polymer will withstand that particular temperature. So at 90 degrees Celsius, if your polymer PVA will be soluble and you can prepare a scaffold. But then if you are incorporating any other bioactive molecule or signaling molecule or anything into the scaffold, those molecules won't withstand that particular temperature. So you have to choose polymers accordingly. So, and during processing or sterilization of an implant or any biomaterial also, you will either sterilize it under autoclaving, heat sterilization, or you will do UV sterilization. You will want to know if that your particular material will be stable at extreme temperature. Then only you will be able to sterilize your material and finally use it for an implant inside the body. So you will want to know these particular temperatures of the polymers that you will be using to prepare your scaffold. That is one reason. Another reason is that so you are using two polymers, same example, polymer P1 and polymer P2, and you are two polymers. Both these polymers will have two different glass transition temperature. So when you are blending these polymers together and when you are preparing one particular polymer, that is one particular scaffold or a hydrogel material, that hydrogel material as a whole will have a different TG, glass transition temperature. So when your TG is between these two TGs, then only you will confirm that you have prepared a hydrogel by combining P1 and P2. Otherwise, it means that you have just blended both these polymers and you have not prepared a completely cross-linked hydrogel. So to chemically characterize your hydrogel also, we will be looking into these characteristics. Am I clear? Any doubts? Just the two reasons to sterilize, to see the stability of your biomaterial construct in different temperatures and to see that your hydrogel has fully been formed and it's not just a combination of two materials. So these are the two main reasons. So the answer is option A. Moving on to the next question. The significance of bioactive materials include dash. So select all the options that apply. Option A, host integration. Option B, bioresponsiveness. Option C, hinders tissue growth. Option D, rendering antibacterial activity.
A and B.
Yeah, host integration, bioresponsiveness, that is correct. Along with it, D is also an option. It will render antibacterial activity. So, bioactive materials usually that can be incorporated in a scaffold or a hydrogel are, for example, nanoparticles. So, you would have heard about iron oxide nanoparticles, ferric nanoparticles. Ferric nanoparticles are usually incorporated in scaffolds or hydrogels to produce antibacterial activity. To render the whole biomaterial antibacterial in nature. So for that, iron oxide nanoparticles are used. And other than that, many phytochemicals that are being extracted from plant samples, they also will be incorporated into our materials to render antibacterial activity. So all these are also called as bioactive compounds incorporated materials. Okay, the next question. Hydrogels are insoluble due to the presence of cross-links. Is it true or false?
True.
Yeah, it's true. So the only reason they are insoluble is due to the cross-linking that we are using here. So the answer is option A. What percentage of water is to be constituted to the total weight of the material to be called it as a hydrogel? So you all might have had this doubt. Why, if it is a hydrogel, then how much should contain within itself? What percentage of water?
At least 10%.
Yeah, that's right. So at least 10% of your hydrogel or the particular material that you are preparing should contain water. So if there is not even 10%, then your hydrogel will definitely not swell in body conditions when it is kept incorporated or grafted in our body, it won't swell at all. So if it is not swelling, then it won't. One main reason is that there won't be release of any bioactive molecules if it is incorporated in the hydrogel. Another reason is that if it is not swelling, then if it is not having pores at all. So if it is not having pores, then it won't have any space for cell migration, proliferation, or any of the cellular activities that a normal ECM will support. So these are the two reasons. So at least 10% swelling, 10% swelling should be present in your hydrogel construct that you prepare. So the answer is option B. So I have given a picture here. Here we can see 3 different hydrogel samples, PAA, polyacrylic acid, 15, 20, 25. So these are the different percentage of acrylic acid that is used for cross-linking the polymer. So these are the 3 different hydrogels, and you are able to see the different swelling ratio for these 3 hydrogels. So what is the correlation between these two? What is the correlation between different polyacrylic acid percentage and their swelling ratio? Yeah, it's very simple only. It's just based on the cross-linking. Can anyone try answering? Hello? Okay, I'll explain. So PA 15, 20 and 25 are different concentrations of cross-linkers that are used in the particular gel. So when you are using a less concentration of cross-linker and when you are able to form a hydrogel, the hydrogel network will be less dense. Yeah, so if this is a hydrogel network and if you are using only 15% of cross-linker, it will be like this. There will be a lot of space for it to absorb water and swell. So as in when you increase the polymer cross-linker concentration, the polymers will be more tightly cross-linked with each other, and thereby we'll get a hydrogel with less swelling percentage. So this is the difference. And you can also modulate the cross-linker concentration based on the percentage of swelling that you require. So percentage of swelling will depend on tissue to tissue. If you are preparing a bone tissue, you will not need a very high percentage of swelling. But then if you are preparing any other tissue, bone or cartilage, you won't need high swelling percentage. But if you are preparing any other muscular tissue, you will need high swelling percentage. So accordingly you can vary your cross-linker concentration. Yeah, so this is the correlation between these two. So at least 10% of water should be in your hydrogel to call it a hydrogel. Okay, so this is all with the questions for this session. I just have a paper that I want to discuss with you regarding the video content, week 2 video content. It'll just take 2 minutes. So this is a paper based on Okay, the paper is titled Intravascularly Infused Extracellular Matrix as a Biomaterial for Targeting and Treating Inflamed Tissue. So, this is a paper from Nature Biomedical Engineering Journal, and in this particular article, what they have done is they have prepared a hydrogel with extracellular matrix. So, you would have studied about decellularized extracellular matrices. So usually they'll take a particular tissue or a whole organ itself. If, for example, a kidney, you would have studied the decellularized kidney in the reading material which was suggested in the first video ending. So in that particular paper, they would have taken a kidney, a whole cadaveric kidney. They would have decellularized it, that is, they would have treated it with detergents, and that kidney they would have then again reperfused it with living cells to prepare a live tissue. So this is the process of using the decellularized ECM. So here what they have done is they'll take tissue, either it is a skin tissue, liver or heart or a bladder or brain tissue. They will cut it into pieces, they will pulverize it, decellularize it, and they will do enzyme digestion, and the final decellularized ECM that they obtain, that they will fabricate into a hydrogel. So that is what they have done in this particular article. So the methodology that they have followed is, so here I have taken a myocardial tissue, which is a heart tissue. So they have taken left ventricular myocardial tissue. They have cut it into fine pieces. They have decellularized it. So if you decellularized any tissue, whether it is an animal or a plant or any tissue, it will be wiped off of all its pigment and cells. They have decellularized it with 1% SDS, sodium dodecyl sulfate. They have lyophilized it. So we studied about the lyophilizing process, freeze-drying process. They have totally lyophilized this ECM and they have brought it into this powder form. And they have again liquid digested this ECM. So after liquid digestion, they have got two fractions, low molecular weight fraction as a supernatant and high molecular weight pellet as a high molecular weight pellet. So they have taken this particular portion, these two fractions, they have separated these two fractions, left and right, and they have prepared a hydrogel. So this hydrogel is prepared just by normal self-assembly, physical cross-linking mechanism. So just by self-assembly, they have prepared a hydrogel and they have injected that hydrogel into a animals. Subcutaneously, they have injected that hydrogel, and after injection also, you are able to see that this hydrogel is still stable, intact, without disintegration. So this is the hydrogel that they have prepared by using decellularized tissue pieces. So this is one article which was like an example for the video contents that we studied in week 2. So you can go through this article. It has been used for treating of inflamed tissue inflammation. Yeah, thank you everybody. That is it for week 2. If you're having any questions, any doubts regarding the week 2 videos or the current session, you can ask me.
Ma'am, can you please explain the question where it was given about the graph for the Young's modulus versus temperature?
Oh, yeah, one second. Yeah, is this the one?
Yes, ma'am.
Okay. So In this question, what they have asked is to find what this point A is. So this point A, when you characterize a particular material, when you keep it in a furnace, when you increase its temperature, it will undergo weight loss. So any material will undergo weight loss when it is kept in a furnace. So the first major weight loss that occurs is at the glass transition temperature. So at glass transition temperature, a material which is ductile in nature will be converted to a material that is brittle in nature. So that particular temperature is called as glass transition temperature. So the elasticity, the elastic modulus also will decrease if it is converted into a brittle nature, and then finally it will be very less when it is in a viscous form, that is at a very high temperature.
So ma'am, you say that it will change into brittle in nature, right?
Yeah.
So ma'am, the point where it is written as rubber seems. So ma'am, what about that point? Will it still be brittle in nature?
Okay, that is just to indicate the flow, like it'll have flow properties. So brittle in nature in the sense it will be very rigid. The molecules or the atoms that is present in that particular material will become very rigid, and when you heat it above that, so when you take a glass and when you are heating it above a particular temperature, the glass which is a brittle component will transform into a viscous component, a component which has flow characteristics. So you will see that in a glass, right? Similarly, it will happen for materials also, all the other materials, biomaterials. So the flow property, they are representing it in that term.
Right.
So when you want to you would have heard about this glass blowing section. So when you want to transform a particular glass material into another shape, you will increase the temperature, you will impart flow, like it will, when it is having the flow characteristic, that time you will change its shape, you will blow it into a different shape. So that is it.
Okay.
Any other questions about? If you are having any questions, you can post it in your forum, the NPTEL forum. I can see that also an answer. I will be sharing this presentation and video recording also with the NPTEL team, and they will share it with you so that you can refer it for further assignments. So this is week 2. This is the second live session. So next week I will be discussing about the week 1 and 2 assignments both. If you are having any doubts, shall I discuss about that, or shall I discuss any other questions?
Ma'am, can we do both?
Sorry, can I?
Sir, can we do both? Like, you will explain us about the week 1 and week 2 questions and some extra questions too.
Okay, fine. Yeah. Then I'll prepare the presentation accordingly.
Thank you, ma'am.
Okay, so if there are no more questions, we can end the call here and we can meet next week.
Thank you, ma'am.
Thank you. Thank you.